All right, everyone. Welcome to our first talk of 2025. And we're very, very happy today to have Sion. And Sion is a fourth year computer science undergrad student at UC Berkeley and Innovative Genomic Institute working in the lab of Professor Jennifer Doudna. Previously, he was a student researcher at Microsoft Research with Dr. Kevin Young and Dr. Judith Amaris, and a visiting researcher at Stanford University with Professor Brian He. His research interests broadly focus on computational system and protein biology, with an emphasis on designing trustworthy methods to learn from mechanistic inquiries of biological process. And let's please welcome Sion to talk about his recent work on olfactory receptors.
Thanks for having me. I'm excited to share a little bit more about some work I've been doing with Judith Amaris and Kevin Yang, which started as an internship project at Microsoft Research. And yeah, so the motivation sort of for this problem was just trying to sort of understand olfaction more as sort of being the least understood of all senses. So deep learning has sort of brought these sort of large gains to sort of our digitization and understanding of vision and hearing. So we have these very interpretable maps for color, and we have pretty interpretable maps for hearing through frequencies, but we don't really have sort of an easy indexable sort of embedding space for something like odors. And so motivated sort of by some early work out of Google Brain, where they sort of proposed some of the earliest ideas of sort of a latent odor map through using graph neural networks, we were sort of interested in building of sort of more biologically inspired, sort of easily indexable, searchable map for sort of discovering new scents and designing new scents. And sort of why sort of choose to work on olfaction? Why is this important or like worth listening to? I think some reasons why I found it really interesting were sort of as I was sort of reading more about it and sort of diving more deeply into it, a lot of sort of vital parts of your health, memory, and emotions are sort of mediated by smell. One example is your sense of taste is a joint combination of the taste receptors and the smell receptors from the air while you chew. Your memory can often be evoked by unique smells from years ago. On a more clinical or health basis, diagnostics have been sort of heavily revolutionized by smell. So there's a lot of work going into Parkinson's and cancer diagnostics because it's been found that Parkinson's patients emit unique volatile organic compounds. And more from a sort of fundamental understanding, more recently, a lot of people who have COVID have sort of lost their sense of smell. And so trying to understand why that's happening is also of interest. And so what was most interesting and probably most relevant to this audience is that it's governed by the wonderful world of proteins. Our sense of smell is really sort of not super well understood. Like for context, the actual discovery of the olfactory receptors and the organization of the system was only sort of awarded with the Nobel Prize in 2004 by Buck and Axel. So we don't really have clear measurement tools for indexing how receptors code for smell yet. And so in essence, the way our sense of smell works is we have these 400 olfactory receptors that each human has with a large degree of genetic variation. So when you smell your morning coffee, a group of sort of many different odorant molecules go into your nasal cavity and activate a different combination of olfactory receptors, which then give your brain a signal, and that gets processed and is what actually results in sort of your perception of a smell or a unique percept. And so these olfactory receptors are expressed in over 1 million olfactory sensory neurons in sort of this small patch of tissue called the olfactory epithelium. And each of these olfactory sensory neurons will express only one olfactory receptor. And so each set that will express a unique OR, when activated, will send all their signals to a unique and common olfactory bulb. And that's what actually triggers a unique signal, which goes to the olfactory tract to your brain. And so this is kind of interesting because it's one of the few sort of clear verticals for signals that actually go straight to our brain. And what's most interesting about this is based on similar advances in our ability to model vision and hearing, and sort of looking at this highly abstracted model of olfaction, we were really interested in trying to build sort of a largely biologically inspired sort of mechanistic model for predicting the end sensory result of a molecule by predicting the actual receptors it activates. And so this is sort of our main motivation is how can we sort of understand the biology by basically first, given a set of different odorant molecules, embed that in some way using geometric learning, try and predict which olfactory receptors it binds to and activates. So sort of a classic protein ligand binding problem, and then use the synthetic predictions from a model on which receptors are activated in conjunction with the molecular sort of embedding from a GNN, and use that to try and predict the perception of a given odorant. And so going through sort of a brief outline of how we thought about this, we first wanted to build some kind of model that uses binding predictions to predict percepts. So we sort of went about trying to build some of the first open source data sets of this problem, and then built a binding predictor that can model OR odorant interactions. We then built a model that can model olfactory perception on top of this, and then we conducted some early interpretability studies in trying to understand if the model is capturing real signal. And so sort of recapping this underlying complexity and why it's a really tough problem that we haven't cracked, essentially different odorant molecules, you have thousands of odorant molecules in conjunction and large mixtures, which are what trigger unique smells. But even on the individual level, an individual odor molecule activates some subset of different olfactory receptors, and that code is kind of what directly triggers for a percept. But instead of something like RGB, where you have different combinations of red, green, and blue, creating a unique color, here you're kind of dealing with many different combinations of sort of 400 choose K receptors, which create really large combinatorial space of different smells. And to make things harder, we know very, very little about olfactory receptor structure. So for example, for context, the first human olfactory receptor was crystallized in early 2023 by a group at UCSF, which is this OR51E2 bound to this fatty acid molecule called propionate. Here, what's interesting is that you really have a key couple of residues that are making in both the transmembrane and extracellular loop that make interactions with this molecule to actually induce binding and activation. And so building a model of this that can capture this locality was something that was kind of interesting to us, and we'll talk about later. Other challenges that we sort of kept in mind in sort of reading about this work is that humans can perceive the same molecules extremely differently. So really small single nucleotide or single amino acid substitutions in olfactory receptors can cause pretty drastic changes in odor perception. So one of the classic examples from our paper in PNAS in 2007 is this OR7D4, which when you basically have a single sort of amino acid substitution from threonine to methionine, it causes like some subset of the population to perceive this molecule androstenone as being pleasant instead of sweaty. So you get a really large variation in the perceived intensity of a compound. And building on this, more recent work has shown that looking at another example of a compound and receptor pair, isovaleric acid, sort of when bound to OR56A4, has a large variance in sort of their pleasantness when sort of conducting human trials and asking humans to rank them based on sort of genotype genetic variation. And so all of this sort of confounds and makes the sort of problem of getting a digitized map for smell much more difficult, and sort of are things to keep in mind when trying to build data sets and models for this problem. And so one major issue on top of the problems with sort of modeling receptors is that if you choose to sort of skip modeling molecular receptor interactions and train models that can map molecules to their sense, it isn't sort of a very simple function of structure. So for example, there's a lot of these sort of triplets of molecules where you have a dissimilar structure, and then you have a very similar structure to your original molecule. And very, very highly structurally similar molecules can have very different percepts or scents. And so this is another part of the motivation of trying to understand the receptor code for us. One of the biggest sort of prior work in this space is this team at Google Brain, now spun out into a company called Osmo, that built some of the original work in training these message passing neural networks on large data sets of molecules with known percepts from fragrance databases, actually. So they're treating this as like a multi-label classification problem, and then showing that they can build a model that can predict the scent of a molecule more accurately than the median human panelist. And on the experimental side, when talking about matching olfactory receptors to odorants, one interesting work in 2019 from Shriram Kusuri's lab at UCLA, now at Octant, actually built a large multiplexed assay for olfactory receptor odorant interactions across dozens of different ORs and hundreds of molecules. And this actually comprised a large chunk of our training data set for this model. On the computational side, there isn't too much prior work in this field. One of the original papers from about 4 years ago, they train basically this SVM where they train an SVM on each olfactory receptor. So each receptor is sort of treated as like a binary task of predicting binding. And they train these SVMs on a bunch of chemical, sort of physiochemical features from RDKit, and then use this model, generate a bunch of predicted features for each human olfactory receptor of sort of binary, does it activate it or not, and then concatenate those to the chemical features and then use that to sort of predict the olfactory perception of an odorant. But some of the issues with this work is that it uses sort of an individual classifier for each receptor, so it doesn't generalize to sort of new receptor space or say non-sort of mammalian or non-human receptors. And it's also not sequence specific, so you can't really deal with sort of genetic variation well. And so sort of given the prior work in the field, we first focused on sort of curating a data set for this problem. And so the dream data set for a problem like this is we want sort of to treat this as an end-to-end learning task where we have a set of odorant molecules, we have their known sort of olfactory receptor code or some subset of receptors that they activate or don't activate and label data for that. And we then predict what they might smell like using labeled data from a fragrance database. And so we can train an end-to-end model on these two tasks. But when we compiled a bunch of molecules for both tasks and tried to plot their Tanimoto similarity, we saw that there was like little to no overlap between the data sets. So there's very few, if at all, any sort of exact structure similarity matches if you look at Tanimoto similarity between molecules that we have receptor data for and molecules that we have percept data for. And even if we use something like some threshold to consider molecule in data set A and molecule in data set B as being the same, one of the big issues with this is that very, very small perturbations to structure can cause a large change to the percept, as we've talked about before. So we kind of had to sort of model these problems separately and then join them later using synthetic data. And so on the percept side, we can actually create data sets from perfumery materials. So there's these large data sets called Good Sense and Leffingwell that are curated by the fragrance community. And they basically contain for each individual molecule known perceptual descriptors called percepts that are constructed by these repeated ratings from fragrance experts. And so this, when we sort of grabbed all this data and merged it into a single data set, we saw that it sort of ended up with it being a very imbalanced multitask data set where if you take a histogram of, say, the molecules per percept, and then the number of percepts that fall into that sort of molecule bin, you have basically 5 or 6 percepts that have 1,000 or more molecules, and then hundreds of percepts or descriptors that have less than 30 molecules. On the receptor side, we found this prior work from a group in France that constructed this data set called M2OR, where they basically construct a data set of about 46,000 olfactory receptor molecule pairs, where they have basically data from the public literature of OR odor interactions and protein ligand binding assays across both human olfactory receptors as well as primates and mice. But here, one thing we had to keep in mind of was sort of more sort of common types of data imbalance where most of the data was non-responsive and there's only a small percent that actually is an agonist or active compound that activates a given olfactory receptor. And most of the data as well is coming from these primary screens where we're testing a compound on a protein at sort of with one injection and at a single concentration. And only about 11% is actually dose response screens where we're testing a compound with multiple replicates and multiple concentrations. And so given this, we first sort of established some baselines and tried to replicate some of the work from the Google Brain team, where we first train a very simple sort of GNN for molecular property prediction, where it works as sort of a GCN that you could then uses some aggregation function to become like to get our dense vector that we then sort of do property prediction on. And so here we sort of found our baseline. Then when we tried to do the same thing for receptors, where we sort of tried to replicate the SVM work, but with GNNs, what we did is we took this M2R data set, which had about 1,237 receptor sequences, and we tried to basically canonicalize them, where we ran BLAST against UniProt to find basically their common OR family and subfamily, and sort of turn this set of sequences which also consists of different variants of known ORs down to about 500 like canonical receptors, where 400 of them are coming from human ORs and 100 are coming from primate or mice. But this sort of expectedly didn't perform very well because each receptor ends up having very scarce data, maybe like a dozen compounds for which it has protein ligand binding pairs, and it doesn't really address any variants or sequence specificity in the model at all, and there's no prior for protein sequence or structure. And so we then started to think more intentionally about how we wanted to model sort of this OR odorant complex. And then on this side, we sort of said, what inductive biases can we use for something like molecule receptor binding? One big thing is the receptor molecule complex. So can we basically provide some details about structure of both the protein and molecule? So in the context of something like OR51E2, the molecule propionate is basically making contact with a couple key residues in the transmembrane domain and the extracellular loop. And so there's some essence of sort of locality where we want to upweigh a key number of residues that are actually key in making an interaction and binding, and sort of identify the binding site. But again, we have very little structural data for this. So we're trying to do this with only protein sequence and 2D molecular structure. And so when we thought about this, as many, I'm sure this audience is super aware of, we started thinking more about sort of protein language models, which we know can really learn structure from unlabeled sequences really well. So PLMs can sort of be trained to reconstruct a lot of sequences through mass language modeling. And sort of in the process of doing this, they implicitly capture early secondary structure and different evolutionary constraints, which sort of infer, which can infer structure from. And sort of we started thinking about using PLMs like ESM2 or Percept. And here we saw that there was a previous paper from the same year we started this project in iClear, where they basically used PLM aggregated embeddings from Percept in sort of their odorant receptor matching work, where they train this model, they essentially embed the embedding from the classifier token and concatenate that to each of the node embeddings in their graph neural network, and then sort of use that to do predictions. But one thing we found that was kind of missing here was that, as people know from work like Chai-1 and ESM-fold, they get a really strong boost from using per residue embeddings as input for structure prediction. And so we kind of wanted to find some way to use the actual per residue embeddings so that we could sort of better specify the binding site to the model and making a prediction about whether sort of compound A binds to protein B. And so here we sort of tried to, we tried to build sort of a perceiver like cross-attention module for basically learning importance scores for each receptor and odorant to actually weigh and sort of aggregate the backbone embedding from our graph neural network and from ESM2. So in this case, given say modality 1, which is like the per atom embeddings from a GNN, and modality 2 is the ESM2 per residue embedding, we basically fuse both into a joint embedding dimension and then use a cross-attention block or two cross-attention blocks where the query for the first cross-attention block is coming from the protein language model. And then in the second one, it's coming from the GNN that represents the odorant, and then the key and the value come from the opposite modality. And so from there, we get these updated per atom and per residue embedding sort of tensors that we can then collapse down to being a fixed size of the number of atoms, the number of residues using a linear layer. And this sort of becomes our importance scores for weighing our original representation. So in essence, when bringing this all together, we take the odorant molecule and we take the olfactory receptor, and we basically feed them both in to ESM2 and the GCN and get two 2D tensors. We use the importance weights from the learned pooling block and then do a matmul to collapse basically the original per residue or per atom embeddings down to a fixed size of just number of basically the dimension size. From there, you can concatenate the two and feed them into a set of MLP layers and basically predict binding. And from here, we ran a bunch of ablations to basically figure out which parts of the model were working well and not. We had a weight loss that was basically upweighing specific compounds that are seen with very few pairs of proteins in the training data, and we did the same for receptors that don't have many odorants. We also did a data imbalance weighing scheme and added that to basically a loss. We also tried comparing whether we compared it against the baseline of just the mean aggregated ESM embedding and the mean aggregated GNN embedding, and using those two to do predictions. And then we also tried other baselines like in compound protein interaction modeling, like perceiver CPI. And the main things we saw were that using a per residue embedding from a pre-trained PLM was sort of gave us the biggest boost in performance. And then from here, now that we sort of had a model that could basically predict OR odorant interactions reasonably well, we got kind of interested in sort of unifying and baking in this essence of combinatorial coding in the model using synthetic predictions about receptor activations to try and model perception. And so here we basically said, can we predict which receptors a molecule activates? And then from doing that, does that actually help the ability of our model to actually predict a molecule's scent? So how this worked in essence is we would take an odorant molecule, we would essentially generate predictions against all receptors in public databases for ORs. The first database being HORD, which is this paper from actually 2 decades ago that mines the entire human genome and finds all probable olfactory receptors and pseudogenes. And so comprises of about 845 different proteins. And then the original training data set from M2OR, which is about, I think, 1,237 proteins, and then generate predictions for the two, and then concatenate basically this vector of binary activations for which receptors are activated by the input molecule. And we use those as supplementary features in addition to sort of the dense vector for the molecule to try and predict which subset of 152 different descriptors a molecule smells like. And here, what we did is we tried to see if you scale the number of receptor sequence predictions you provide as context, how does that sort of improve the prediction power? And one of the things that was kind of cool about this is that this also allows us to take basically the predictions, so say each of the receptors that we're generating predictions with, and then introduce sort of genetic variation. So we can like introduce mutations to different receptors that have been found in the literature to cause a large difference to smell or to the percept of a molecule, generate the predictions, update basically the predicted activations, and then generate predictions to the smell and see if sort of our percept code is actually modified as a function of the receptor code, basically, and sort of measure these variant effects. And so now bringing all this together, since we had sort of this model that can do, we had a data set and we had models that can do OR odorant interaction prediction and can then build on that model to do olfactory perception modeling. Could we do some initial work on interpretability where we try to apply these models to reveal interesting receptor percept coding patterns? And then maybe in the future, use this model to mine and deorphanize olfactory receptors with no known ligands, and then experimentally maybe validate them. And so the idea here was basically, given sort of this data set of molecules and percepts, so around 6,000 different molecules with known descriptors of how they might smell, let's use sort of the model that predicts receptor activations to generate synthetic predictions, and then cluster each set of molecules according to their percepts and see if there's consistently some receptors that get activated for, say, a unique smell. And so our ability to do this, we found, was like largely a derivative of how broad a given percept family is. So the idea here was, given sort of all odorants, you drop all odorants that bind to more than 30% of olfactory receptors according to the model. So any promiscuous odorants, and you do the same for receptors. So we end up dropping about 25 receptors that sort of are predicted as sort of broadly activating a large chunk of the odorants in our fragrance databases. And here what we see is when you sort of look at an odor family that's a lot more specific, like all molecules that are described as smelling like meaty, as being roasted or nutty, we find like some with pretty high signal, like consistently a couple of receptors that seem to be predicted by the model as being active. Whereas if we take something that's more broad, like fruity molecules, we tend to find a lot more variability in sort of which receptors are activated by which molecules. And on the other side, an experiment we were really interested in running was basically testing this idea that humans have evolved a ton of olfactory receptor pseudogenes. And this is kind of theorized because we've sort of developed a decreased reliance on our chemosensory perception for survival compared to like dogs. And so we wanted to see if our model could cluster and discern binding patterns by different OR subtypes, or by whether a protein was an intact OR that could actually be activated, or a pseudogene. And so on the left, what we looked at is we clustered all the olfactory receptors by OR family in this mined OR subgenome data set, and then ran predictions on all molecules in the good sense Leffingwell data set. And we found something that was pretty interesting here, where we have some specific families that nearly consist of only predicted orphan olfactory receptors. And so there's a lot of receptors with truly like uncharacterized functions that are maybe highly tied with pseudogenes. And so what we did on the right is we then decided to cluster them by whether they were a protein coding gene, so something that can actually be activated and isn't misfolded, versus something like a dysfunctional OR pseudogene that's maybe being truncated. And so here what we found is that if you take the median, basically, number of ORs activated by a molecule that are functional versus a pseudogene, you get basically per molecule that on average 3 functional ORs are activated compared to 1 for the pseudogene. So the model is sort of accurately able to not predict a bunch of binding in sort of these pseudogenes that really shouldn't be binding to any odorants, or maybe a much smaller subset of odorants. So this is pretty interesting, and could be sort of an interesting way to maybe use the model to mine more and more receptors and find sort of new olfactory receptors. And so some of the takeaways, I think, with this work on a high level is that sort of this combinatorial coding problem is really complex. And we need techniques to bridge sort of the sparse pairwise data we have for odorant molecules and receptors to actually decipher like unique, clear patterns for combinatorial coding and actual underlying scents that are triggered by a subset of, say, 3 or 4 receptors in tandem. And so this is both sort of driven by experimental work and more say multiplexed assays, and also more clever techniques like optimal transport to sort of make up for the lack of data between different modalities. And also that in sort of incorporating inductive biases from leveraging these pre-trained models can actually really help us get over this data limited regime by sort of really making use of their evolutionary and structural prior. And yeah, the overall sort of goal of this work was to sort of try and take a couple of steps towards building a really strong, an initial sort of end-to-end map for olfactory information flow that we can sort of use as a tool for interpretability and olfaction, and also to potentially do the design and discovery of new synthetic odorants instead of having to use natural products. And yeah, some next steps with this work that we're thinking about is sort of integrating more pre-trained models that sort of use a lot of the graph transformers that can do molecular property prediction quite well. And then also studying the receptor map further to see if it can capture unique, different novel hierarchies of OR coding. One thing that I'm really excited about looking into is sort of studying the underlying representations learned by these models and seeing if they capture like the natural statistics of odors. So there's some work from the Osmo team showing that their sort of principal odor map, or their underlying sort of representation from their penultimate layer of their GNN, can actually tend to organize around metabolic activity. So for example, it can essentially, it's highly correlated with, say, multi-step reaction paths. So like looking at these multi-step reaction paths form a smooth trajectory in their latent space. And they also are highly correlated with different metabolic reactions and the odors they emit. And so there's some interesting experiments in studying metabolism and olfaction using these models. And then also on the sort of more prediction side, using curriculum learning between the two tasks to sort of improve generalization, and also using more synthetic 3D structures to train a future model using models we have now like Chai-1 or AlphaFold 3 or both. And yeah, that's everything. Thanks for listening. And I'd like to thank my mentors, Judith and Kevin, for sort of introducing me to this world and both advising me on sort of the more olfaction and neuroscience and machine learning for protein engineering side, as well as the MSR New England Lab and undergrad program for their support. And yeah, I'm happy to take questions.
Thank you so much for this amazing talk. And you can unmute yourself or just post your questions in the chat. I'm going to allow everyone to unmute yourself. There's 2 questions in the chat. Maybe we can start there.
I can answer the first question. The first question is asking if there's any overlap between train and test sets such as Horde, and whether there's how much data leakage we could expect. I think this is a good question. The main sort of clarification here is that Horde as a data set is just sort of a database of olfactory receptors. There's sort of no known data for all of these receptors for what odorants they actually activate. And so I'd expect a good chunk of them to be receptors that we see at training time on many different odorants, but the actual Horde data set isn't actually consisting of different odorant receptor pairs. So I don't know if that helps answer that. And then on the other side of like generating novel compounds, I think the next step would be basically seeing if you can invert a model like this, where sort of use this as like a sort of a classifier guidance type approach and build a generative model that is sort of guided by this the latent space of this model to actually generate novel compounds. So I think that would be like the natural next step for this, is sort of turning it into a generative model.
Do you guys plan to have any experiment validation on this, or that's going to be another new next step?
I think that would be a next step, is like working with experimental collaborators who are more on the neuroscience or more on the sort of high throughput screening side, on like sort of downstream data sets that the model hasn't seen. But that would definitely be like probably a follow-up work.
There's a new question asking from an application point of view. Do you generate the models of receptors and their ligands?
Um, I'm do you know if you could clarify your question a little bit more? Like, are you asking whether we're using like synthetic data as input to the prediction task, or like the receptor odorant task, or I'm not like 100% sure what the question is asking.
Yeah, feel free to unmute yourself
Yeah.
and just ask a question.
Does the model learn the yeah, so the first model basically tries to learn sort of the binding preferences of different olfactory receptors and ligands. And then the second model uses the predictions from the receptor model. Is like we run predictions across all known human olfactory receptors, and then use sort of the vector of sort of predicted activations as additional features to try and predict how a molecule might smell. Yeah, the last question is asking, from the ablation studies, it seemed like the per residue PLMs had a big impact on performance. Did you guys explore different architectures downstream of those PLM embeddings at all? This is also a good question. We didn't play around with changing the embedding. What we did play around with different ESM model sizes, and we saw that basically up until 650 million, we saw an improvement, but when we scaled to 3 billion, we saw like a drop off in performance for matching receptor odorants on our test set. So I think beyond a certain point, it seemed like scaling and using a larger ESM2 model didn't help beyond 650 million. So we mostly stuck with 650 million after that.
Did you try doing any, like when you report like the AURC, did you think about the train test split and like how you make sure that you don't have too much similarity between them?
Yeah, the main thing we did with the test split is because so much of the data is coming from noisy primary screens, we have sort of a minimum threshold for how much of the test set is coming from dose response screens. So we make sure that at least 25% of the test set is coming from actual dose response screens that are actually tested with multiple replicates and at different concentrations to sort of avoid sort of a false positive rate. And then we also play around with the weights of different samples based on the false positive rates of primary and secondary screens for each sample.
Thanks.
I think there's a new question in the
Cool,
chat.
yeah. So we can generate a structural model of receptor and ligand, right? That would improve the interpretability and help in. Yeah, I think so. There's a couple things we've been doing on this end that we're hoping to have in the updated manuscript. We've sort of been looking at sort of, for a lot of examples, we've sort of seen if we can say, for example, where we actually have crystal structures, can we sort of look at the attention maps, where we get these attention maps that are sort of number of residues by number of atoms, and we also get the learned sort of weighted importance scores. And we've been trying to look at whether the model's sort of importance scores are highly correlated with like the binding site, for example, or like actual atom level interactions. So that's like something we've been doing to see if the model's sort of weights or the model's attention maps are actually learning to sort of learn which are likely contacts, or for example, if it's actually focusing primarily on parts of the transmembrane domain and the extracellular loop where binding is actually happening. Cool.
I think there's recently a very interesting trend in enzyme design that people start using coevolution for substrates. I was wondering if it can be the case when you modeling with your ligands, you can have any like coevolution signal that either protein side or on the ligand side that might help you improve the model performance.
Yeah, I think it might be interesting to see if like given that there's some like constraints on coevolution for class A GPCRs and olfactory receptors in general, like sort of constrain evolution, actually baking those in as like sort of like an initial template for where to sort of pay attention to in the per residue embeddings would be interesting. That's like something that could be an interesting way to sort of see if like fixing the initial weights is actually helps the model a little bit. So it doesn't prioritize things that are sort of far away from the binding site. Or like the general scaffold of or like the general cage of the protein.
Yeah, if there's no other questions, let's thank again. Oh, there's one more question. Okay.
Yeah, so the question is asking, can your model offer an interpretation on how combinatorial binding of a ligand to mutated receptors causes different odor perception? So I think, yeah, so it can offer a prediction. I don't know if like the predictions, and obviously all of these are predictions, and so I think we can like use them as like initial inquiries or like initial hypotheses, but we always have to validate them. But yeah, in theory, what you could do, and one of the nice things about like a model like this is you can basically take the same like a known ligand receptor pair and just introduce a bunch of different mutations to the receptor and then generate predictions for each of the different variants and see how that changes both the underlying receptor binding code and then the downstream like scent code or like percept code, basically. So that part is kind of a nice feature of it.
Maybe we can wait 1 or 2 minutes to see if there's any other questions. There's a new question. Did you try specifying sequence regions like TM regions for binding and see if your model improves?
We did not, but that's a great suggestion of like, yeah, I think one good idea would be just like fixing in the aggregation function, like setting some constraints for or like a mask on where the attention scores should be above 0 or which ones are like, yeah. So I think, or like above some threshold would be good, or like a good way to basically specify like using the residue embeddings from the TM region. So that's a good idea.
All right, I guess that might be the last question we have today. And let's thank again for this amazing talk, and thanks Seon for giving talk today, and thank everyone for coming to the first seminar of the year, and we'll see you guys again in 2 weeks, and Happy New Year.
Thanks for coming. Take care, guys.